I'm putting together this short video out of concern that students are having some difficulty with this kind of question. I've been grading the discussion forums and I'm consistently simply seeing students replicate the sequence and not actually carry out gene expression. So I want to walk through this problem with you. Here I'm asking you, what is the product of gene expression for the following sequence in a bacterial cell? And I give you some sequence. Now, after reading through the textbook and hopefully watching the lecture videos that were provided underneath your objectives on the Chapter 7 page, you recognize there's two steps involved in gene expression. The first of those steps is called transcription. And the second step in gene expression is called translation. So transcription, if you'll recall, is converting that information in DNA into RNA. And the message that we're going to read to be able to produce a protein is called messenger RNA, and it's abbreviated to mRNA. The second step of gene expression is translation. It is using the RNA that is produced in transcription as a code, a template, to produce a protein. So our final product in all of this is going to be a protein product. Now, some other things that we need to look at before we can actually address this question. Well, we should recognize at this point that proteins are produced by structures known as ribosomes. And ribosomes, if you'll recall from Unit 1, come in two sizes. They come in a 70S size and an 80S. 70S ribosomes are found in bacterial cells, but they are also found in eukaryotic mitochondria. ADS, on the other hand, are the ribosomes that are found in the eukaryotic cytoplasm. So they're found free floating in the cytoplasm, but they're also found associated with the endoplasmic reticulum that we refer to as the rough endoplasmic reticulum. Now, looking at this question, you will have genetic code or a copy of the genetic code on your test. So it's not a matter of memorizing this thing, it is a matter of knowing how to use this thing. So let's take a look at our question and begin to break this down. So the first thing that we need to be able to do is take a look at the question, see what we're asking. We're asking for gene expression. So we're ultimately going to produce a protein product when we're finished answering this. Secondly, what kind of cell are we doing this in? I've asked you to do this gene expression in a bacterial cell. So this will incorporate the use of a 70S ribosome. Now, the reason why this is important is as we do this protein production, you're going to find that when we get to what is referred to as a start codon, it's called AUG, these two different ribosomes interpret that information differently. 70S ribosomes, when they reach a start codon, will put into place an amino acid known as formal methionine. ADS, on the other hand, will put into place an amino acid called methionine. These are two structurally different amino acids. You can find them on page 205 of your textbook to take a look at their structures. All right, so 70S, when we see a start codon here, the AUG, we're going to put into place formal methionine. It's abbreviated FMET. The other thing we want to take a look at, what kind of code have we started with? Well, seeing some T's in there tells me that this is a DNA sequence. So I'm going to start off with DNA. That is first going to have to be transcribed into an RNA message for the ribosome to read. So when we transcribe DNA to RNA, your thymine will pair with an adenine on the mRNA, adenine with uracil, cytosines with guanine, and guanine with cytosines. So you just continue to carry out that process. And there's your mRNA sequence. So this is what we are going to read. These are codons. So each set of 3 is a separate codon. And that's what we find in the genetic code is we see the codons represented from the mRNA message that's read by the ribosomes. Now, reading that genetic code, let's take it one codon at a time. When we look at a codon, it includes 3 separate nucleotides. The first nucleotide of a codon, we find it represented on the left-hand side of this genetic code. The second codon is across the top, and the third one is on the right-hand side. So when we take a look at this codon number 1, our first nucleotide is adenine. So we look along here and we find the adenine. Adenine, all of the codons that are in this third row all begin with A. Then we look at our second nucleotide, it's U. So we look across the top, we find U, our uracil. Uracil is all represented as a second nucleotide in the first column. So we're going to stay in this box. In the third row, first column is where we're going to find our codon. If we go across the right-hand side, we find this G in this third row. And we come across, and right here is our codon of interest, and it codes for, it says on your chart, methionine. But we just discussed that when you have a 70S ribosome, instead of placing methionine as the first amino acid, this ribosome will put into place formal methionine. That is important that you pay attention to that. We're working with more than one type of organism in this course. Now let's move to the second amino acid, second amino acid coded for by the second codon. This second codon is CGG. So we do the same thing. We locate our C, find our G, move down. So we see it right here. CGG codes for arginine. And I'm just abbreviating here. They won't be abbreviated on your exam. Third codon codes for UUU. So we find U, U, and U. There's our phenylalanine. And then our fourth codon is UAC. That would be tyrosine. All right, so when you're finished doing those steps, knowing what to look for as far as the answer, well, our question asks, what is the product of gene expression? And the final product of gene expression after doing the 2 steps is a protein. These are the amino acids that will make up the protein that we're looking for. So this would be the answer that you'd look for on your exam. Now I would like you to take this information, how this was done, and take a look at the questions that were provided for you in your discussion forum and attempt to answer those. Provide me with some feedback, see if you can tackle those now, and then we'll try a couple more of these together so that you can do well on the test when you see these kind of questions. Good luck to you all.